Okay, so thanks very much for the introduction and the invitation to speak here. And the title's already been announced. So I'm Nathan Lapierre. I'm a PhD student at UCLA, and this is our metagenomic profiling method. So just to cover the terminology here. So actually, as Nina kind of just alluded to, there's two questions you can ask with microbiomes, which is sort of who is there, what organisms are present, and what are they doing, what's their functional role. So last talk, we got a little bit of the functional stuff. I'm going to be focusing on who is there, so what organisms are there. So this metagenomics term refers to whole genome shotgun sequencing of microbes recovered directly from their host environment, which could be some soil, ocean water, human gut, whatever. Microbes are everywhere. And the benefit of this approach versus traditional culture-based approaches is that in a traditional culture-based approach, obviously, well, first of all, some organisms are difficult to culture, or if not impossible, in the lab. And also doing that doesn't capture the full complexity of real-world communities. So that's metagenomics. And then the profiling part of it is predicting the organisms present in the sample and their relative abundances. So you want to generate something like this, where you say E. coli is 30% of the sample, S. aureus is 12% of the sample, and so on. And specifically, I'm going to be focusing on reference-based profiling, which means we have a database of known reference genomes of microorganisms, and we get the sequencing reads, and we use the database basically to construct this profile. And obviously, the most sort of classic way you can think of doing that is to just do alignment, most classically BLAST, I guess. And the obvious benefit of that is, people generally consider BLAST to be highly accurate. It's intuitive and easy for non-technical people to understand. And if you return alignments, they're useful for a lot of other things. You can do other things than just profiling. But the drawback is that this can be prohibitively slow. So that's basically why people have to develop a lot of methods now. It can take months to blast samples. And another method is MEGAN, which is based on alignments that you can produce with BLAST, or this protein alignment method, DIAMOND. So more recent approaches have been developed that deal with this efficiency issue. So one of them I call K-mer matching. There's a lot of different ways you can use K-mers, but I'm referring to kind of the way Kraken-2 and Clark. And these methods tend to be very fast and have high recall, but because they're constructing a big data structure of all the K-mers in the sample, that has to be stored in memory. They tend to require high memory usage, and they've been shown to demonstrate a lot of low abundance false positive organisms in the sample. And then another type of approach is to only use marker genes. So marker genes are supposed to be specific genes that help you differentiate between different organisms. And so these methods, a couple of examples are metaflan2 and MOTUS2. They tend to be high in precision and have very low memory usage. And they're faster than just alignment, because usually you're just aligning only to specific marker genes. But some of the drawbacks are that sometimes there's low recall, and sometimes the abundance estimation is off because you don't get sort of the information from being able to align to all parts of the genome. You're focusing on a specific subset of genes, and it's not easy to retrain the database. So if somebody else has a custom database that they want to put in with a certain set of reference organisms, it's usually not easy to do that with these methods. So this is just kind of an overview of a lot of different approaches to the problem. So some of the challenges this brings up, I've kind of mentioned some methods have high recall or high precision, but it's rather difficult to balance precision and recall. So intuitively, you can think it's easy to have good recall because it's easy to kind of just take your reference database and say, okay, almost all of these organisms are in the sample. It's also easy to have high precision because it's easy to just say confidently one or two organisms we're highly confident in are present in the sample. So a challenge is trying to balance both of those. And sort of attached to that, or relatedly, can we achieve the accuracy and balance of precision recall of alignment-based methods without the prohibitively slow running time or memory usage? So our hopeful answer to that question is yes. And this is where our method comes in, Metalign, which is efficient alignment-based profiling. And the intuition behind our method is that the main limitation to alignment is slow runtime due to massive reference database sizes. So you're taking all of these sequence reads, and as we know, modern sequence reference databases can have hundreds, be hundreds of gigabytes. So performing alignment against the entire reference database is very slow. So if we can quickly filter out organisms from the database that clearly aren't present, we can dramatically reduce alignment time. So there's a lot of organisms you should be able to sort of quickly see there's nothing in this genome that is present in the sample, so you can quickly rule it out without having to perform a full alignment of every read against that genome. So that's the intuition. So in Metalign in stage 1, we do that database pre-filtering. In stage 2, we perform alignment against a subset that database that we get from stage 1. And then in stage 3, we deal with multi-aligned reads, which are reads that align to multiple different genomes, and we compute the output profile. So that's sort of the overview. Now I'm going to go through each step of that. So the first and probably most important step is our pre-filtering stage. And we use this containment min-hash concept, which is called SMASH. That's how you pronounce that. So SMASH uses K-mer sketches to estimate the containment index, which you can think of as the percentage of K-mers in an organism that are present in the reads. So it's related to the Jacquard index, which is the proportion of K-mers in the set over the union of two sets. So a method that estimates that is called MASH. So that's where the SMASH name comes from, except it's containment index. And it's showed by our co-author here, David Kozlicki, in the paper that introduced the containment min-hash concept, that this Jacquard index is biased towards 0. And so the containment index is preferable when you have a large difference in set sizes. So intuitively, you can imagine if we're testing some genome from our reference database and we want to see if they may be in the sample, we measure we have them get the K-mers in each. However, there's the number of K-mers in the full set of sequence reads is much, much, much larger than the number of K-mers in a single microbial genome. So calculating the Jacquard index over the entire union of both sets tends to be biased towards 0. So containment index, again, is instead just is sort of the percentage of the K-mers in an organism that are present in the reads. So the denominator is the number of K-mers in the organism. Okay, so, and by doing this, we can use a really comprehensive reference database in an efficient amount of time. In our case, we use all NCBI assemblies, which is REF-Seq, GenBank, and everything else we could find. And I mentioned before that this mathematical concept of containment min-hash was introduced by our co-author in this paper, but Metalign is the first paper that really puts this into practice in any application, but in this case for metagenomic profiling. So again, we're going to test each genome, and SMASH calculates a containment index, which is 0 to 1 percentage of K-mers from the genome that are in the reads. And we take all organisms above a certain containment index cutoff and create a subset database of those organisms. And in practice, this really dramatically reduces the size of the database. So this often reduced our full database, which in our case was 243 gigabytes gzipped, to about 0.5 to 5 gigabytes, which is, it varies because it depends on the diversity of the sample, how many organisms you need to include in your database. But the point is, it's a dramatic reduction, and then alignment is much faster. We use Minimap2, which I assume most people are familiar with, but it's basically a more recent alignment that's very fast and very accurate. We compared it in some tests with BWA MEM and found that it was just as accurate for us. And so we do that alignment, and then we produce our output profile. Okay, but after we do alignment, we have to, and before we get the profile, we have to deal with these multi-aligned reads. So these are reads that align to multiple genomes. And so they're ambiguous, but we found that if you just throw away those reads, then this substantially worsens your abundance estimation, because those reads tell you certain subsets of genomes that have shared reads. So there can be very many of those reads, and if you throw them away, then you're losing information. So to deal with this, we applied a two-stage approach. It's pretty simple. Basically, we first calculate the relative organism abundances using only the uniquely aligned reads, and we save the multi-aligned reads and which genomes they map to. And then after we've calculated the unique read organism abundances, then for the multi-aligned reads, we proportionally assign them to organisms according to their unique read abundance. So to make that concrete, let's say we have 3 reads here and 3 genomes. And according to the unique reads, genome 1 is 50% of the sample, genome 2 is 20%, genome 3 is 30%. And so our sequencing read 3 here maps to genomes 2 and 3. So these two, and out of that set, genome 2 is 40% of it, 20 out of 20 plus 30, and genome 3 is 60% of it. So we assign 40% of that read to genome 2 and 60% to genome 3. So I guess the thing to note here is that we're not explicitly performing read classification necessarily. We're just trying to generate the most accurate abundance estimation possible. And then I sort of parenthetically noted here that we also deal with all sorts of things that can happen with paired end reads, like one end maps and the other one doesn't, or one end maps to one organism and then the other end maps to multiple, and so on. So we deal with those, but I've left out the boring details of that. Okay, so we deal with these multi-aligned reads, and then we produce our output profile. And so just to give you the full picture here, again, we have our input sequencing reads, our reference database. SMASH quickly tests each genome and gives us the containment index for all the genomes. And then based on a cutoff threshold, we include organisms in this subset database, which tends to be much smaller than the full database. And then we perform alignment from the reads to that database, and then deal with those multi-aligned reads and produce the output profile. So that's sort of the sketch of the meta-align method. Okay, so now that you have a sense of the method, I'm going to go through the results. So just quickly, evaluation metrics, probably most people are familiar with these, but just to recap. So precision and recall measure false positive and false negative rate respectively. So precision is true positives over true positives plus false positives, and recall is the same except false negatives. So your precision and recall are high if you have few false positives and false negatives respectively. And then the F1 score, again, is this key issue of balance of precision and recall, and it's the harmonic mean. So the F1 score basically is high if the precision and recall are high and also close to each other. So it really emphasizes balance between the two. So that's for presence absence performance, and then the L1 error is for abundance estimation performance, and this is basically just the sum of absolute value errors between your predicted and actual abundances. So if we predict an organism is 10% of the sample, but it's actually 60%, that gets an L1 error of 0.5. And I should note in the following results I'm about to show, because precision and recall F1 score are 0 to 1 scale where higher is better, whereas L1 error is a 0 to 2 scale where lower is better, we flipped the L1 error and rescaled it from 0 to 1 so that higher is better to get everything on the same scale. Okay, so these are the evaluation metrics and the data sets we tested. So people in metagenomics have probably heard about this. There's this big benchmarking competition called Critical Assessment of Metagenome Interpretation, which came out a few years ago in Nature Methods. And they simulated some really realistic data sets that had high diversity and high missingness, by which I mean there were novel strains comprising large percentages of the reads, and novel strains meaning they simulated novel strains that aren't in any reference database. So it's you can't possibly have them in your reference database. So kind of simulating a realistic scenario. But of course, simulations always only go so far. So with real data, there's some noise that we have to deal with. One of the nice things you can do is use a mock community. So a mock community is a microbial community specifically designed and created in the lab. So it's still real data. They actually create the organisms and sequence from them. So it still has that aspect, but you know the ground truth. So you can actually measure your performance. And so we use this Peabody et al. Mock community. And this community in particular was a low diversity community. So it only had, and diversity, but basically I mean how many different organisms are in the sample, and relatively well-known, well-characterized organisms, and not a bunch of missing data. So these two situations are very different. So we wanted to see if methods were consistent across these two different scenarios. Okay, so now I'm going to get into the results without further ado. So these are the results on the CAMI1 challenge data sets. These are genus level results averaged over all 8 data sets. So I mentioned there's 8 data sets and they have varying community diversities. So on the left here, we have a heat map with precision recall F1 scores, scaled L1 error. Again, higher is better. This is a 0 to 1 scale, so the lighter cells are better. And basically and here I have a precision versus recall scatter plot. And basically you can clearly see 3 methods: Metalign, Megan plus Diamond, which I mentioned was one of the alignment methods, and MOTUS2 did really well. Other methods, I mentioned Bracken, Kraken2, and Clark, these Kamer-based methods. I should mention here that we applied a 0.01% abundance cutoff threshold, meaning we excluded organisms with below 0.01% abundance, which is kind of necessary to be fair to these methods, because they generate like thousands of these extremely low abundance organisms. So to be fair, we excluded those, but there's still low precision. And then some of these other methods, again, I mentioned Markov gene-based methods like Metaflan2 have had high precision, but poor recall and poor abundance estimation. So, but Metalign, Megan, and MOTUS2 did really well. But now if we look at the time in memory, on the left here is wall clock time in hours, and on the right is peak memory usage. This is over all 8 data sets. So this is the peak at any point while processing all 8 data sets. This is the wall clock time summed over all 8 data sets. And so we observed that Megan plus Diamond was by far the slowest method. These were all run with 4 threads, by the way. So note that this is a log scale here in hours, so it's a couple order of magnitude slower than the rest of the methods. So this is over 1,000 wall clock hours to process all 8 data sets. And Kraken2 is extremely fast, but most methods were around 10-ish hours, so a little over an hour for each of these data sets, which, by the way, were 50 million paired end reads each. So Metalign is kind of in the middle of the pack here. We're not trying to be the fastest. And in terms of memory usage, Clark used by far the most of any method, over 150 gigs. I mentioned some of these Markov gene methods, for instance, Metaflan2 and MOTUS2 have extremely low memory usage, just about 2 gigabytes. And then Metalign is kind of in this middle pack of methods that used about 40 gigs. So overall, Metalign is kind of middle of the road in timing and memory. We didn't focus on those. We basically wanted to get the most accurate results possible while being reasonable in these. But Megan was very accurate, but much slower. And then, so that's the CAMI data. So now this is the Peabody mock community data. Here, so I mentioned this abundance cutoff threshold. So I wanted to vary that because that can also, as I mentioned, that can affect the performance of the methods. So you can see here Clark and Bracken have a really low F1 score because they have a lot of low abundance false positives when you apply no abundance cutoff threshold. But actually, when you apply a reasonable one, they perform pretty well. Notably, MOTUs 2, which did really, really well on the CAMI data, was generally the worst performing method on this data set by a wide margin. And Metalign, when any abundance cutoff threshold is applied, was the highest performing method, and actually achieves a perfect precision and recall, so no false positives, no false negatives, with an abundance cutoff threshold of 1% or 5%. These values are higher here because there's only, I think, 8 or 9 genera in this data set. Bracken actually also achieves a perfect F1 score at 5% abundance cutoff threshold, but it's more sensitive to that parameter. Okay, and this is at the genus level again. Finally, there's another thing you can ask, which is basically, is Metalign was it just more accurate because we used a more comprehensive reference database? And I mentioned this issue of being able to retrain a method using a different reference database. So Kraken 2 actually makes that pretty easy. So we basically took our reference database, which is very large, and retrained Kraken 2 with that to see if that would improve its performance, and it did. So again, this is back on the CAMI data. So its precision obviously dramatically increased, as did its F1 score because of the improved precision, and its abundance estimation error also improved a lot. So it does help a lot. It was still a bit lower in precision and F1 score than Metalign. So it did help a lot. However, in terms of timing and memory, it's still extremely fast. But using this really comprehensive reference database, Kraken 2, its memory usage went up to 325 gigabytes, which is obviously that's hard on a lot of users. So it becomes infeasible for many users. Okay, so just to recap all of this, Metalign was the only method that performed well on both data sets with reasonable runtime and memory usage. That was our goal. The non-alignment based methods generally struggled in one setting or the other, and most non-alignment methods had low precision or recall. Megan, which is the other alignment-based method that we assessed, was comparable in accuracy to Metalign, but the runtime became infeasible with large data set sizes, and retraining Kraken 2 on Metalign's comprehensive database got much better results, but the memory usage became infeasible for many users. So the key takeaways I wanted to emphasize from this are that Metalign is able to achieve highly accurate results by leveraging this alignment-based approach in combination with a really highly comprehensive reference database. And so obviously alignment is pretty trusted, and highly comprehensive reference database is good. The question is, how do you do that while having a good runtime and good memory? We saw the issue was runtime. Kraken with that database was the issue was memory. And the way Metalign's able to maintain solid performance there is due to this SMASH-based pre-filtering stage. So the pre-filtering, as I mentioned, dramatically reduces the database size, so that kind of solves the runtime issue. And then SMASH is based on these lightweight K-mer sketches, so the data structures that have to be read into memory are pretty small, even for a really huge reference database. So that's how we're able to do reasonably in runtime and memory. And so that's it. I'd like to thank you all for listening. I'd like to thank my co-authors, David and Sergey, co-supervised the project. Eliezer is my PhD advisor, and Mohammed helped out a lot with the custom database experiments. We have a paper up on bioRxiv. We're hoping to have it in a journal pretty soon, and we have a GitHub that's up. Metalign is also available on Bioconda, so you can use it from there. Oh, and I'd also like to thank Lana, because she helped a lot with improving the writing in the manuscript. And that's all. Thank you very much, and I'd be happy to take any questions.